Hi, we are team number one. Our team is composed by me, Mauricio Alcalá, Jorge Moeller, Vela, Fernanda Partida, Moya, Valeria Zambrano, Ramírez, Brenda González, Garza, and Husseini, Challa. And we're going to explain all the topics that we saw in stage one. So first, I'm going to start with the introduction, that is biology and its field of action. It says basically what is biology, the branches of biology that are anatomy, anthropology, botany, embryology, biomedicine, biochemistry, cytology, genetics, histology, immunology, virology, and zoology, among other things. So then we have 1.1 that is characteristics of living beings. So it says that the characteristics that unify living beings can be summarized in next points. First, we have organization, that says that in living beings, the components are organized complexly. Life on Earth is characterized by being organized and hierarchized in levels in which the lower ones give sustenance to the superiors, and nothing can exist or function without this complex organization. The second one is the genetic code. It says that it is a sequence of nitrogenous bases that combine to produce words in a language understandable to all living beings, so it is considered to be universal. Then we have response stimuli, and says that living beings can respond to different stimuli such as light, sound, temperature, chemical substances, among others. Then homeostasis, that is the fourth one, and it says that is a process of self-regulation phenomena that lead to the conservation of steadiness in the composition and properties of the internal environment of an organism that allows it to have a free life. The fifth one is metabolism, that says that metabolism of living beings use energy to maintain homeostasis. That means they must take components from the external environment. Metabolism involves the destruction of molecules, that is catabolism, and the formation of new molecules, that is called anabolism. Then we have growth, and it says that all living beings, to a greater or lesser extent, increase their volume, that is, they grow, everybody. Then we have reproduction. It says that to perpetuate their species, individuals must reproduce themselves. Here we have asexual reproduction. Its main characteristic is the fact that the descendant is genetically identical to its predecessor, since in this modality sperm and ovules are not involved, and therefore there is no genetic variability. Sexual reproduction, that is this type of reproduction involves the fertilization of two different cells, that is the sperm and the egg. And parasexual reproduction. Through parasexual reproduction, genetic variability is achieved through the exchange of DNA that is carried out by three types. Conjugation, that is the donation of genetic material through a breach or pilus and its incorporation in the genome. Transduction, that is a transmitting agent like virus, carries DNA that is introduced into the genome of the host bacterium. And transformation says that a bacterium that induces and incorporates DNA fragments from that bacteria into its own genome. And the last one characteristic of living being is adaptation ability and evolution, and basically says that the world had changed and changing and will change. Every one of the species settled on this planet is forced to adapt and evolve in order to survive. If it doesn't happen, the punishment will be the extinction.
Hi, my name is Husseini Challa, and I am going to talk about the history of the microscope, the cell, and the cell theory. To understand how humanity has increasingly expanded its knowledge about cellular characteristics, We must identify the instrument that allows the observation of structures invisible to the human eye, the microscope. Who invented the microscope? It was invented by Zacharias Janssen in 1950, Galileo Galilei in 1609, or Cornelius Drebbel in 1619. The first person who names this invention as the microscope was Giovanni Fieber in 1625. According to the records of the time, the magnification range achieved by this microscope was 3 and 9 times. It is very probable that Janssen did not foresee the potential of his invention for the science. Who was able to glimpse its importance and use it for the scientific purposes was Robert Hooke in 1665. It was Hooke who linked the history of the microscope with that of the cell. Years later, Anton van Leeuwenhoek developed a new technique to manufacture lenses that allowed him to observe samples with magnifications up to 200 times. He published his results in 1684. History of the cell. The official story begins with Robert Hooke, who in 1665 published the observations he made with the microscope he created. Hooke observed the bark of the cork oak. He gave up to these species the name of cells, from the Latin cellulae, that is, little chambers. Then, years later, Anton van Leeuwenhoek was able to describe and observe in more detail cells, such as spermatozoids and protozoa, and some kinds of bacteria. Then, Theodor Schwann and Matthias Sladen proposed the first postulate of the cell theory. The cell is the elemental unit of plants and animals, and it is the essential basis of life processes. Then, Robert Brown described the nucleus in 1831. Johannes Evangelista Purkinje introduced the term protoplasm. That is, he was the first to describe the cellular cytoplasm in 1839. Meanwhile, Rudolf A. von Kolker identified the mitochondria in 1857, and Rudolf Virchow proposed that all cells come from pre-existing cells in 1858. The work of Louis Pasteur on the metabolism of yeast and ascipips, 1860, cleared the way to the progress of research. August Weismann discovered that both current and ancestral cells are similar in structure and composition in 1880. Cell theory. The first one is that the cell is the morphological and structural unit of all living things. In other words, all living beings are made up of cells. The second one means that every cell comes from a pre-existing one. The third one is the cell is the psychological unit of life. The fourth is the cell contains the hereditary information. And it can be added a fifth postulate, that is, the disease begins in the cell. Thank you.
Hi, my name is Jorge Moeller. I'm from the classroom 218 and I'm going to talk about the cell. Well, what is a cell? Cells are complex thermodynamic structures that use energy to maintain the conditions that make possible their existence. The structure and function of a cell has the following three characteristics: the plasma membrane, the cytoplasm, and the genetic material. The plasma membrane defines the boundary between the cell and the environment that surrounds it. It is the structure that allows it to have individuality, and even if it belongs to a whole or not. Cells have DNA, which is the genetic material in which the information is contained so that the cell can be functional and survive. They contain all the metabolic machinery to fulfill their physiological functions. And within the limit imposed by the plasma membrane is contained the cytoplasm in which the organelles are immersed. There are two types of cells, prokaryotic and eukaryotic. First, we're going to talk about the prokaryotic cell. The prokaryotes are cells without a nucleus. The most noticeable difference is that their genetic material is found in the cytoplasm, in an area called the nucleoid. They are also unicellular and can be reproduced asexually or parasexually. The components in all prokaryotic cells have a plasma membrane, a cell wall, a cytoplasm, a nucleoid, the ribosomes, prokaryotic compartments, and according to the type of the prokaryote cell, they can have flagella in the outer membrane, periplasm, capsule, cytoplasm inclusions, pili, glycocalyx, biofilm, S-layer, plasmids, and mesosome. In the other hand, the eukaryotic cells it has two types, the animal and the vegetable. The most notable difference among them is the presence of the nucleus, the membranous compartment in which the genetic material is confined. Animal cells are structurally more complex and they present distinctive features that define them. That is, the animal cells are different in their components concerning the vegetable ones. The components of the animal cells are the plasma membrane and some membranous organelles, which are the nucleus, the rogue endoplasmic reticulum, the smooth endoplasmic reticulum, the Golgi apparatus, the lysosomes, peroxisomes, the mitochondria, and some not membranous organelles are the cytoskeletal elements, the centrosome, the centrioles, ribosomes, and cytoplasmic inclusions. The plant cell has components similar to those of the animal cell because both are eukaryotic cells. The organelles that have a similar composition and function in both cells are plasma membrane, nucleus, cytoskeleton, Golgi apparatus, ribosome, endoplasmic reticulum, mitochondria, and peroxisomes. So, the organelles and structures that are found in plant cells are cell wall, central vacuole, plastids, chloroplast, leucoplast, chromoplast, amyloplast, elioplast, those are plastids, and it also has glyoxysome and plasmodesma.
Hi, my name is Fernanda Partida and I will explain cellular transport. Everything that enters or leaves a cell has to pass through the cell membrane, which is an organelle that is present in all cell types. The cells need the nutrients or molecules required to carry out its metabolic activities to enter the cytoplasm, and also that the waste or the product it generates, such as hormones, come out of it. Transport can be classified according to the amount of molecules that pass through the membrane. If they pass one by one, then the transportation can be active or passive. Active. This transport spends energy and goes against gradients of concentration. Passive. This transport does not waste energy and goes in favor of concentration gradient. It can be simple diffusion, facilitated diffusion, and ultrafiltration or dialysis. Simple diffusion. The molecules cross the lipid bilayer. Gases such as carbon dioxide and oxygen pass through it freely. In facilitated diffusion, the passage of molecules depends on the presence of the integral membrane proteins that allow this passage. These proteins can form channels or complex transporters. Ultrafiltration and dialysis. Water and some solutes pass through the membrane due to hydrostatic pressure. The flow is always from the highest to the lowest pressure. This is in the case of the glomerular filtration in the kidneys to form urine. When the transport through the membrane is massive, then it is classified as endocytosis and exocytosis. Endocytosis is incorporation of material into the cell, and it is divided into three. Pinocytosis. This concept is comparable with the drinking of the cell since the cell incorporates molecules in suspension. Phagocytosis. This process is comparable with the eating of the cell because through it the cell incorporates large quantities of materials and even whole cells. Receiver-mediated endocytosis. In the cell membrane, there are receptors to which specific molecules bind, which are then incorporated into the cytoplasm. Exocytosis, secretion of the material produced by cells. It is divided into two. Regulated. In this case, the cell responds to a signal to release its products. Constitutive. The secretion is constant. Levels of organization. The components that make up living beings and the living beings themselves constitute levels of organized complexity. The first level is that of the atom. The atoms form molecules and biomolecules. The biomolecules constitute the organelles and structures, the set of organelles, to form the functional unit of life, the cell. A set of cells specialized in a specific functions form a tissue. The tissues form organs, the organs are grouped into systems, and in a multicellular organism, the systems constitute the individual. Now let's see the levels that go beyond a single individual. A population is made up of individuals of the same species. The community, or biocenosis, is formed by a set of populations. The grouping of populations of all domains, together with the abiotic factors, physical environment, constitutes the ecosystem. Finally, the biosphere is the total set of ecosystems present in Earth. So it goes: atom, molecule, cellular organelles, cell, tissue, organ, organ system, organism, population, community, ecosystem, and biome.
There are four types of tissues. The first one is the epithelial tissue that covers the surface of the body and the inside of some organs. The second tissue is the conjunctive or the connective. This tissue that is filling between organs and connects with other tissue. The third tissue is the nerve. This is constituted by networks of neurons. And the last one is the muscular tissue that thanks to this tissue, humans can move their muscles. Now I'm going to explain the main systems of the human body. The first one is the cardiovascular. This system transports oxygen, nutrients, and other substances to the cells. The second system is the lymphatic. This system defends the body against infections and diseases. Then is the digestive system that processes food and absorbs nutrients, minerals, vitamins, and water. Then is the endocrine system. This system provides communication within of the body through hormones. Then is the integumentary system that provides protection against injuries and loss of fluids. Then is the muscular system that provides movement, support, and heat production to our body. The next system is the nervous. This system collects, transfers, and processes information. Then we continue with reproductive system that this produces gametes that are sex cells and produces sexual hormones too. Then the respiratory system that delivers air to places where gaseous exchange occurs in our body. Then is the skeletal system that supports and protects the soft tissues of the human body. Then is the urinary system that eliminates excess water, salts, and waste products of blood and body. And this controls the pH of the body. Then is the immune system. This system defends against microbiological pathogens and other diseases.
Hi, I'm Valeria Zambrano, and I'm going to explain what does homeostasis mean. It basically says that homeostasis is a property of cells, tissues, and organisms that allows the maintenance and regulation of the stability and constancy needed to function properly. Homeostasis is a healthy state that is maintained by the constant adjustment of biochemical and physiological pathways. An example of homeostasis is the maintenance of a constant blood pressure in the human body through a series of fine adjustments in the normal range of function of the hormonal, neuromuscular, and cardiovascular systems. These adjustments allow the maintain of blood pressure needed for body function despite environmental changes and changes in a person's cavity level and position. Other homeostatic mechanisms, for example, permit the maintenance of body temperature within a narrow range. Homeostasis is brought about by a natural resistance to change in optimal conditions. An equilibrium is maintained by many regulatory mechanisms. All homeostatic control mechanisms have at least three independent components for the variable being regulated: a receptor, a control center, and an effector. The receptor is the sensing component that monitors and responds to changes in the environment, either external or internal. Receptors include thermoreceptors and mechanoreceptors. Control centers include the respiratory center and the renin-angiotensin system. An effector is the target acted on to bring about the change back to the normal state. At the cellular level, receptors include nuclear receptors that bring about changes in gene expression through upregulation or downregulation and act in negative feedback mechanisms. An example of this is the control of bile acids in the liver.